Sorry about that. I forgot my free screencaster only goes up to 15 minutes. So let's try this again. Um, so we're talking about the enzymes that bacteria secrete, and that these enzymes are part of their normal metabolic pathway. So this is something that the bacteria is just kind of going about its daily life, going, okay, I'm going to convert these nutrients to this because this is what I require. And they're secreting these enzymes. But sometimes these enzymes can cause disease in the host. And we can use these enzymes also as a way to kind of help us identify which microbes are present within an individual as well by using um, different cultures. And that helps us to characterize some of these microbes because of these enzymes that they secrete as part of their normal metabolism. Um, so exoenzymes. So exoenzymes, as it says, a lot of times can damage host tissues. So we've got a couple of different examples here. It's like mucinase, which can digest a mucus coating, and that can help them to become established and adhere to a host cell. Hyaluronidase, as it says, digests hyaluronic acid, and that helps to hold cells together. And so if hyaluronidase is present, it can digest that connection between the cells. And in some ways, that allows the bacteria to get even further, deeper into tissue, but it can certainly destroy tissue when that is released. Coagulase versus kinase, so whether they form blood clots or the exoenzymes help to dissolve clots. So coagulase is something that will allow your blood to clot, so they can secrete chemicals that act as coagulants. And so having the blood clotting, they believe, may help to disguise the bacteria sometimes, so it may aid the bacteria because your immune system may not be able to find it if it's kind of surrounded by a lot of fibrin formed by the blood clotting. And this is what occurs with Staphylococcus aureus. Staphylococcus aureus is one that will secrete a coagulase, and we actually get to see this during the lab time, see that kind of clotting occurring. Versus kinase, which dissolves clots. And this can also aid in the diffusion of bacteria through the body. So if your blood is not clotting, then it can allow for rapid diffusion so the bacteria can get into your bloodstream much more quickly. So besides exoenzymes, we can have what are called exotoxins. So toxins are molecules that in small amounts tend to generate basically a range of effects within the host tissue. It can damage host tissue. It could suppress an immune response. And exotoxins tend to be produced by both gram-positive and gram-negative bacteria. And they can have a wide range of effects on different cells. So they're namely, the toxins are kind of named after the type of cell that the toxin targets. So we can have what are called neurotoxins that affect the nervous system. You can have enterotoxins that target the GI tract. You can have hepatotoxins that affect the liver, nephrotoxins that damage the kidneys. And here we have an example where we'll be seeing this in lab. You're seeing a blood agar plate on the right-hand side there that's divided up into 3 different quadrats, and you're seeing 3 different bacteria that have been just kind of swiped across the blood agar plate in those 3 quadrants there. So we're looking at pathogens that are capable of lysing red blood cells is what we're looking for typically on a blood agar plate. So they will lyse the red blood cells to gain access to nutrients. And so they do this by releasing an enzyme called a hemolysin. And so the pathogens that make hemolysins basically lyse the red blood cells in the media, and that tends to generate kind of a yellow zone around the colony. And that's what you're kind of seeing over here, is that kind of cleared area around the colonies because they've been able to lyse the red blood cells. So bacteria that can do this are said to be hemolytic. Now, some bacteria do not lyse the red blood cells. Instead, they just kind of oxidize the hemoglobin, which is they get at the oxygen carrying component of the blood. And so the bacteria with this property are said to be alpha hemolytic, and they turn the blood agar kind of a greenish color. So alpha hemolytic, you get the kind of the greenish yellowish color, which you're seeing over here. Okay, so the beta hemolysis is kind of your complete destruction, where you get the yellow zone, and your alpha hemolysis is where you get the kind of greenish yellow, because it's just destroying the hemoglobin. And then you've got some that do not produce any hemolysins at all, which is what you're seeing at the top here. And we'll kind of go over this in lab once again. Now, all gram-negative bacteria have an outer membrane, and we've talked about that structure, having kind of that additional membrane layer on top of the peptidoglycan layer. So that outer membrane is typically rich in lipopolysaccharides, or what we call LPS. So lipopolysaccharides consist of a lipid portion, which is called lipid A, and then there's kind of a sugar portion, which refers to that saccharide tag of the name. So the lipid A region is basically an endotoxin, and it's toxic to us and to other animals as well. So gram-negative bacteria mainly release endotoxins when they die. So when gram-positive bacteria basically do not make endotoxins because of their cell wall composition. All right. So when your immune system, or let's say antibiotics, kill gram-negative pathogens, then the endotoxins are released into the body, and they can cause symptoms. So a lot of times you feel fever and chills and body aches and inflammation, the nausea. Some of that is coming from these endotoxins that have been released when your immune system kills these bacteria and releases the lipopolysaccharides into your system. Some lipopolysaccharides may also be released as bacteria divide, but that's only a small amount. The great majority comes from when the bacteria, gram-negative bacteria, are being killed off. So these lipopolysaccharides are not easy to neutralize. There's no effective kind of therapy to counter their effects within the body. So it's something what you just kind of have to endure. All right. All right, causing disease. All right. So disease can be caused by the pathogens indirectly. So just their simple presence triggers your immune system, and sometimes by your immune system can respond, as it says, excessively or inappropriately. We kind of talked about that with sepsis when that happens. So sometimes your body kind of goes overboard and that can be the cause of the disease as well, or kind of mainly contributing to it, is sometimes when your immune system goes overboard, and then a lot of bad things start to happen because your body's just attacking a lot of cells. There can also be epigenetic changes. So epigenetic refers to kind of controlling the expression of certain genes, and this can happen when these microbes affect how the genes are going to be expressed, either by using an interference RNA or by attaching chemicals to certain histones, which are found on the DNA molecules that help to regulate genes. And these epigenetic changes can be passed on to other cells after cell divisions. And so the interesting thing about this is that even though the microbe may have been killed off, or the bacteria might have been killed off by your immune system, these changes, these epigenetic changes, sometimes can persist afterwards. So even though you might no longer have a viral load or a bacterial load present, there still could be some of these epigenetic changes that are still regulating the genes that are going to be expressed, and have some longer lasting effects because of that. Talking about different types of infection, so just some terminology. This is the chart from your book. Talking about localized infections. As it says, the microbes enter the body and remain confined to a certain area, so that's why we call it localized. So the infection is just in one location within the body, versus a systemic infection where it spreads to several different sites. So chickenpox, especially one of those, gets into your nerves, your nerve cells. Focal infections, as it says, are infectious agents that spread, and it says they're usually asymptomatic from one site and is carried to other tissues. So periodontal infections leading to cardiovascular consequences, right? So you might have something that you're not even paying attention to in your mouth that's going on in there. We have a lot of bacteria that's in there. And then you get a little cut, and now it's in your bloodstream and traveling to other parts of your body. Mixed infections. We talked about polymicrobial infections. We've talked about secondary infections. Now we got acute and chronic. So acute refers to the fact that this is a very short lived infection, versus a chronic infection that's going to persist for a long period of time. All right, so it says acute would be something like a cold or the flu, versus a chronic infection, which is HIV, which you can have for basically the remainder of your life. You'll have it. Warning signals of disease. So we want to differentiate between the two terms of a sign and a symptom. So typically when you go to the doctor's going to ask you why are you here? What are some of your symptoms? And so you're going to tell them what you are feeling. Those are your symptoms. So symptoms are subjective evidence, as it says. This is kind of what you are sensing, what you are feeling at the time. So it says you may feel pain, you may feel dizziness, you may, you're just this malaise that you feel, versus a sign, which is what the physician is noting. So it's objective evidence. So we can say, well, I'm going to take your vital signs. Is your blood pressure up? Do you have a temperature? So these are objective evidence that somebody else is recording. All right, so somebody kind of outside of the disease who is looking at you and saying, okay, well, I see these things. Okay, and they're able to record them as evidence. Now, sometimes you have asymptomatic or subclinical infections. So you have no noticeable symptoms, even though you have